Hey, how's it going? My name is Gino Gomez. I'm in the Snyder Lab at Department of Genetics, Stanford School of Medicine, as well as Cal State East Bay, Department of Biological Sciences. I wanted to share Human Cell Atlas geographical distribution locations of the members of the Human Cell Atlas, or all across the world. We have scientists engaged in the HCA activities that are they've expressed interest and participate in the HCA. So we can see all the different sites, how much each country contributes to the Human Cell Atlas. These are the different journals that the Human Cell Atlas has published in. And then out in the Snyder Lab, we focus on the digestive system or the gut biological network and the gut cell atlas. We can see here the different organs of the human intestine of the gut, and in the large intestine and colon, we have the different organs: transverse colon, ascending colon, descending colon, cecum, and sigmoid. So those are the different organs that are found in the digestive system. In the histology, sort of like a cross section of uh, of the digestive system, we can see the mucosal externa and the submucosal core, as well as the villi. And then how we see it in sections, we see the crypts and the villi kind of coming up from the mucosal barrier, where the lamina propria are sort of stacks of lamina propria, and then you have the mucosal mucosae across, and then above that is the crypt villi, um, different zones. So the mucosa is the innermost layer. There's a submucosa, mucosal layer externa, and the serosa or the adventitia, which is the outermost layer. We can see in the histology in these slides, the crypts are on the outside, and below that are the mucosa and the submucosa. So the serosa and mucosal layer externa is more striated. And then we have the like these Brunner glands in the crypts as well as the villi. And then inside the villi, we can see lacteal space and the lamina propria, kind of like bringing together the intestine. And then on the outside, you have the absorptive epithelium. And then here's kind of like a cartoon depiction of what the structure of the intestine looks like, where we can see the villi and the crypt, and going down, descending or going down, we have the tip, middle, base, TA zone. TA is for transit amplifying zone, or transit amplifying TA zone, and then the stem cell zone. And in the Peyer's patch, we have the different immune cells. You can see T cells, B cells, macrophages, and dendritic cells. And then in specific, there's like crypt-based columnar cells that are in the base of the crypt, and then they differentiate, or there's the different structures are, or the different cells are the absorptive progenitors and the secretory progenitors. In the absorptive progenitors, where they interact with microbes, you have the enteroendocrine cells. So you have entero and endocrine cells, where we see all different types of epithelial cells, where they differentiate from enterocytes and the M cell. M cells where they interact with the with the barrier, and then in the secretory progenitor, you have the endocrine progenitor, which differentiates into X, K, D, and L cells, I cells and N cells, EC cells, TAC1 EC cells, SCT EC cells, pant cells, goblet cells, sentinel goblet cells that interact with goblet and sentinel goblet cells, and then tough cells as well. So those are all the different secretory progenitors, and the absorptive one is the M cell from the enterocyte. So you can see that there's notch activity in between these cells that really activate the different CBCs. So the structure of the intestine is simple yet diverse, and the mesenchyme the intestinal mesenchyme space is more stroma. Then I went over some of the different signaling proteins BMP, EGF, and BMP-I and WNT went. So these are different signaling genes and proteins that are in the structure of the intestine. So here I was able to map out the healthy intestine, but we'll go over this at a different time. I wanted to show the colon immune atlas. So in the colon immune atlas, we have a UMAP where we can see the different cell types. And in the different cell types, we have the cluster of cells. So there's 12 different cell types that I was able to use LIANA and get a clear map of steady state inference. So we're able to infer the cell types with LIANA Plus, and then get a cell by gene dot plot where we can see the cells and the markers. And then I was able to do feature engineering for the single cells and then map out the INK T cells, B cells, macrophages, monocytes, and dendritic cells, as well as exokines, which are um, about signal moieties that are that are that are signaling moieties that respond to exercise, either autocrine, paracrine, or endocrine signaling. So they can either be with the same cell, the cell next to it, or a cell far away, and they are released in response to exercise. So that's IL-6, IL-7R, and IL-7, and then a housekeeping gene, J chain. So we can see different clusters, these different feature engineered single cell markers with CD1D, and then CD4, CD8, and CD19, CD68, CD14, and CD209. So these are all immune cell markers. But I wanted to focus on the first one here in the top cluster and then the top right cluster, where we can see this CD1D, which is an invariant natural killer T cell. These cells are specialized in innate like T lymphocytes that bridge innate and adaptive immune systems. And this is one of the rare cell types that is in both innate and adaptive immune system. And they're a subset of type 1 NKT cells. So they're defined by semi-invariant T cell receptors, or TCRs, and their ability to recognize lipid and glycolipid antigens presented by the CD1 molecule. And then, unlike the standard T cells, which are adaptive, that recognize foreign proteins, foreign antigen, we have iNKT cells that recognize self and microbiolipids. So the lipids on the microbiome, which could be the cell wall, you can recognize those. So once they're activated, they release the massive amounts of cytokines to jumpstart the immune response. And then the development, so like how do they differentiate? How do they become iNKT cells? In the thymus, there's effector subtypes, so they start expressing specific transcription factors. So for iNKT1, you have a TBET, which produces interferon gamma, and that fights viral infections and tumors. In iNKT2, you have a PLZF, which is a zinc finger uh, transcription factor that produces IL-4, which promotes a Th2 response and promote and you'll have tissue repair and also controls parasites or parasitic infections. And then iNKT17, you have regulated ROR gamma transcription factor, which produces IL-17, which induces expression of Th17 cells and defends against extracellular bacteria and fungi. So those are, this iNKT cell type that is specialized and that is able to work in both innate and adaptive immune responses is really important um, for cancer immunotherapy. So they have unique dual characteristics of T cells and NK cell receptors. And they've been a subject of extensive oncology research. So they have the ability to detect and destroy cancer cells while modulating a broader TME or tumor microenvironment and makes them promising candidates for allogenic cell therapies. And then getting to the extra there's IL-6, which is in the liver, adipose, and muscle, and it's able to allocate energy and oxidize fat, as well as have a systemic response for anti-inflammatory signaling. And then in IL-7 receptor, they're active in the lymph nodes, the thymus, and muscle. So they're also they're also myokines. So they're myokines, adipokines, and hepatokines. IL-6, IL-7, and IL-7 receptor. IL-7 and IL-7 receptor, you have naive T cell survival and immune surveillance, as well as satellite cell regulation. Satellite cells are a special muscle cell type, and their IL-7 and IL-7 receptor are able to regulate that specific muscle cell. And then the J chain, we can see the different clusters. It's housekeeping. We're able to see expression distributed throughout the UMAP. So it's a good measurement, kind of like a housekeeping gene that we're able to measure the J chain, which joins. It's a 15 kilodalton glycoprotein that is secreted in a multimeric IgA dimer and IgM pentamer antibodies, and commonly used in biotechnology applications like ELISAs and Western blots. It's also synthesized as a plasma cell marker. So these are mature B cells that make the same antibody to a specific antigen. And then the secretory component, another function of the J chain, is that it binds to polymeric IgA receptor or Ig receptor, so PIG receptor. And those are in the epithelial. So all the epithelial cells that you've seen they also express the J chain. So that's why you see it all distributed throughout the entire UMAP. And then in the violin plots, we can see which cluster has the most genes. And it looks like cluster 6, 1, 0, and 11 percent of mitochondria. Not that much mitochondria in these cells. IL-6 could be making the most, allocating the energy there. And then in the J chain, we see 0, 1, 2, 5, 6, 8, 9, 10, 11. And then IL-7R, which is the exokine receptor. We have clusters 3, 4, 5, and 7, as well as 6 and 8, and then some 11. And that's in this colon immune cell atlas. So getting back to the UMAP and what heat map, and then as well as looking at the specific genes and the similarities that were expressed in the colon immune atlas with a new tool called Scanpy, a single cell RNA-seq next generation computational tool where we can measure the cell types as well as the RNA transcripts. So which RNA transcripts are being the most expressed? We can see the different clusters here and all the different colors, yellow, red, blue, green, brown, and purple, as well as pink and gray. And then all the similarity genes. So it looks like for orange and yellow, CCL5, HLA-B, RREG, NXA1, IL-7R, CD74, HLA, DRA, as well as IgHA, RPL, RPS, MZB, and some heat shock proteins. And then in the pink, gray, red, blue, and purple, you can see that sort of the same genes and the levels of RNA transcripts expression there. In the cell types, you can see from monocyte to dendritic cell, innate cells, higher activity here. And then in the B cells and NK and T cells, you can see higher expression there. And then some Th1, Th17, as well as activated CD4 T cells. And as well as this UMAP. So UMAP, heat map, and then an RNA transcript from single cell RNA-seq data from the colon immune atlas. And then getting back to LIANA, I wanted to show the donor region and cell types as well as a number of genes. So here are two different UMAPs. And the first UMAP, I was able to get the donor region and cell type. So we have the different donors, mostly 290B, 298C, and 302C, and then the different organs. So we have the mesenteric lymph node, the transverse, sigmoid, and cecum organs. So there's like a cross organ map of the different regions. And then what I had to do is get the cell types, the specific cell types. So we know the downstream signaling, but the specific cell types in this UMAP, we're able to see B cell memory cells. So memory B cells with follicular B cells, and then ILCs, as well as lymphoid DCs, and then CD8 T cells, as well as Th17 cells. So those are close together with Treg cells, and then unique clusters of gamma delta T cells. And then in another UMAP of the same single cell RNA-seq data, we can see the number of genes in clusters here. So that's trying to see the number of genes. And then we're also able to make a cell-cell interaction network plot where we can see the cell-cell communication. And we have here NK cells as well as gamma delta T cells. So the NK cells, we can see mast cells, macrophages, lymphoid, LYVE1 or LIV1 macrophage, and ILC follicular B cells and CD8 T cells, and then PDCs, which are plasmacytoid dendritic cells, and then also GDT, which is a gamma delta T cell. And then we have CDC1, CDC2, which are conventional dendritic cells. And so next we have gamma delta T's, and the gamma delta T cells we can see LIV1 macrophage, monocyte, and gamma delta GDT, as well as cycling GDT, cycling DCs, CDC1 and CDC2, and those are for the gamma delta T cells. For CD8 T cells, I was able to find or generate a network plot where we can see follicular B cells, ILCs, LIV1 macrophage, lymphoid DCs, mast cells, and macrophage. A little bit of macrophage, but more mast cell. And we also have CD8 Ts, cycling GDT, and activated CD4T, B cell IgA plasma, B cell IgG plasma, and then B cell cycling, B cell memory cells, and CD8 Ts autocrine after that. Lastly, I wanted to go over the LRIs, the specific ligand receptor interactions, which we're able to see here in a source plot where we see the ligand receptor interactions and the target cells with NK cells and gamma delta T cells that interact with the source. So we have a source or a sender and a target or what we call a receiver. And we can see the magnitude rank as well as specificity rank. So there's in the monocytes, in the innate immune system, we have S100A8, CD69, which S100A is a calcium and zinc binding protein that's predominantly found in the myeloid lineage, and specifically differentiated neutrophils and monocytes. And that S100A8 is interacting with CD69, which is a vital transmembrane protein that's recognized as one of the earliest cell surface markers in lymphocyte activation. So one of the first lymphocyte cell differentiated, we can see CD69 cell surface marker, and that's found in the monocyte interacting with NK cell and the gamma delta T cell. And then above that, we have activated CD4 T cells, CD8 T cells, and monocytes that we can see HLA-B, which is an MHC complex, and KLR-D1, which is a killer cell lectin type receptor D1 that encodes CD94 protein. And that's primarily found on the surface of the natural killer cell and certain T cells. So that interaction, the interaction HLA-B and KLR-D1 with activated CD4 T cells happens with NK cells and gamma delta T cells. The CD4, the CD8 T cell is also has in it also interacts with NK cell and gamma delta T cells. We see more activation with the NK cells in this interaction, and that's true for the monocytes as well. So those are the LRIs that I was able to find with LIANA, and we can see that in a LRI plot. The other tool that I used was single cell immune receptor, um, where we can see CD3E marker, which is a T cell marker, and we can see the activity on the left side of this UMAP, and a higher amount of activity lower, and there's TCR, T cell receptors there. So the air part of scIR is able to detect the T cell receptors in blue here. And then in orange, you see there's no immune receptor. So there's no BCR or TCR. But in blue, we can see the TCR, and that's in the single cell immune receptor. Lastly, I want to show the QC metrics of filtered samples where we get the log 10 counts in cells for the organs on top and then the cell types on bottom. So we can see that both of these are continuously going up with each region, such as the cecum, sigmoid, transverse, and mesenteric lymph node. And then for each cell type, or like the different cell types that I went over with LIANA. So we can see here, LIANA was able to make the cell-cell interactions in UMAPs as well as ligand and receptor interactions. And then Scanpy was able to decipher the single cell RNA-seq data, just like LIANA, but give us the RNA transcripts in an activity plot, as well as a dendrogram and UMAP. And then LIANA was able to produce cell-cell communication network plots where you can see kind of like in a chord plot the different interactions between the cells, which cell is communicating which cell. And then in the gray plot, we were able to see the activity and the magnitude of the expression for each ligand receptor interaction and cell type, cell-cell interaction with the LRIs together. So kind of combining those two, we're able to see kind of like the most detailed interactions with the LRIs, and then which single cells have immune receptors with single cell immune receptor, single cell scIR, and AIRR for the single cell TCR, VDJ-seq data. So we had single cell RNA-seq data and single cell VDJ-seq data together in the immune cell atlas, where we were able to look at the immune cells in the immune atlas. So thank you for your time. I wanted to go ahead and go over the human intestine colon immune atlas in the human cell atlas today. And thank you for your time. If you guys have any questions, feel free to reach out and email. It's dgomez@horizon@csueastbay.edu. And my personal email, sfdanielgomez@gmail.com. Thank you. And for more information, you can go to the humancellatlas.org and find out what's going on this year. We have 2026 Human Cell Atlas General Meeting that is happening this week. So I wanted to go ahead and contribute by posting a video on the colon immune atlas where we can see sort of distinct immune cell niches in the colon organs. Thank you. Thank you for your time.